So these notes are going to go into the PBS course file. First, you got to make your foldable. So you fold the yellow piece of paper in half. Then open it back up. Then fold over the right-hand edge till it meets the crease. And then fold that over. Then draw a line along that edge. And then draw another line for that section, so you've got about 1/3 and 2/3 there. Draw a line along that edge. Okay. So this is on my left-hand side. This is bacterial morphology, and here is our foldable. So bacterial morphology, there are characteristics of individual bacteria cells. So you have to use a microscope to see them because bacteria are very small. One component of bacterial morphology is shape. So we have bacillus, coccus, and spirillum. These are the three most common shapes, the shapes that we're going to look at. Bacillus, coccus, and spirillum. Bacillus is a rod-shaped bacteria. Coccus is a round or sphere-shaped bacteria. And spirillum is a spiral-shaped bacteria. Another component of bacterial morphology is the fact that we can look at how the bacteria are arranged. The diplo prefix means double or two. The strepto prefix means strips, means chains of bacteria. Staphylo means clusters, kind of like a bunch of grapes. So if I have bacteria that look like this, where I've got two rod-shaped bacteria together, diplo for the two, bacillus for rod-shaped. So now I have two spherical bacteria, so diplo for the two, coccus for the spherical shape. Streptococcus, strep means the strips or the chains, coccus, so I have a chain of circular bacteria. Streptobacillus, I have a chain of rod-shaped bacteria. And then finally, Staphylococcus, Staphylo because I've got a cluster, it kind of looks like a bunch of grapes, coccus because they are circular in shape. So underneath we have the 6 pictures. Cut them out, and then underneath each picture use your bacterial morphology to describe them. Talk about the shape and arrangement of each type. Colony morphology. Colony morphology deals with the colony of bacteria, with all the bacteria that grow together. So colony morphology describes the characteristics of the isolated bacteria colony. So we're not looking at individual bacteria. We're looking at the colony, the bunch of bacteria that are growing together. For bacterial morphology, you need a microscope. For colony morphology, you just look at the plate. So the things that we look at on the colonies are the color of the colony, the form, the elevation, the margin, and the size of the colony. So color is pretty self-explanatory. What color is it? Form, here are some examples of different bacterial colony forms. Most of the ones we look at are circular. Elevation, so if we're looking at the plate at the side, does that bacteria colony, is it raised? Is it convex, so it really pops up? When it grows, does it look like it's flat? Those are probably going to be the 3 that we will have in class, but how does it grow up off the plate? Margin deals with the edges, so smooth edges are entire. Undulate, that's right. Okay, so if they're kind of wavy like that, that's undulate. And then finally, the size of the colony, you'll use an actual ruler and find the diameter of that colony. Colony morphology, we can see it without any aid to the human vision. Bacterial morphology, we need a microscope. So we wrote this before, but we're writing it again. Bacterial morphology is the information on the left-hand side page. It shows the characteristics of individual bacteria cells. That's why we need a microscope. So examples of bacterial morphology are size and shape of the individual bacteria. Gram stain, how do they look after they've gone through a gram stain? And the arrangement, how are they arranged? So size and shape is Bacillus coccus beryllium. Arrangement has to do with are they diploid, staphylococcus, streptococcus, that sort of thing. And then, like I said, we'll do gram stain and look at are they staining and show a gram-positive bacteria or a gram-negative bacteria.